In this video, we're going to be talking about Protistan predators, these single-celled eukaryotes that are preying on other organisms. And last time we talked about primary producers, and we saw that most of the primary production in the ocean is done by fairly small single-celled organisms, so maybe no surprise that a lot of the consumption of those phytoplankton is also by other small single-celled organisms. So we mentioned primary producers, and again, primary production, this is the initial source of organic carbon that goes into the food web. It's done by autotrophs, and in particular in the ocean, primarily it's being done by phytoplankton or photoautotrophs, using sunlight energy to fix carbon into organic food for the rest of the food web. And then consumers are all the other organisms in the web that have to get their organic carbon by eating that which was made by someone else. So we have phytoplankton being eaten by a consumer, primary producer eaten by a consumer, and of course you can have a primary consumer eaten by a secondary consumer, and that's what most of the rest of the food web is. So there are a variety of different types of consumers. We talked about primary producers and then consumers, but how they get that organic carbon can vary. So grazers are a type of consumer that typically we consider them to be eating vegetation, eating a part of another organism, typically a plant, without actually killing it. That would be grazing. Predators are things that actually kill their prey. Symbionts. So symbiosis is this prolonged interaction between two different organisms where at least one of them benefits, and in some cases that will involve one getting food from the other or nutrition out of or from the other. And this can be a mutualistic relationship or it could be a parasitism. Then we have scavengers, which eat things that are already dead, unlike predators, which kill their prey. Scavengers find things that are already dead to eat. And then in the next video, we'll be talking about decomposers, in particular bacteria that do the final stages of degradation of the last bits of detritus and organic matter in the ocean. But for this video, we're going to focus on the predators, and in this case, the microscopic predators. In the last video, we talked about the small scale on which primary productivity is being done. These very small organisms like the photosynthetic bacteria, pico-eukaryotes, somewhat larger coccolithophores, and diatoms and dinoflagellates. We also said that the small things tend to be more abundant than the big things, so a lot of the production is happening on those smallest of scales. And so, not too surprising, you look at the predators that are eating those things, they're just slightly larger than the most abundant phytoplankton out there. So we have a lot of these predatory microplankton swimming around, feeding on the cyanobacteria and these small other phytoplankton. But there are other larger types of phytoplankton like the dinoflagellates and the diatoms. Who's eating them? Well, some animals can eat those, but as you remember, the dinoflagellates we said were a really mixed bag, and some of them are photosynthetic, and we introduced them last time. Some of them are heterotrophic. Things of this same scale, other dinoflagellates of this same size could be swimming around eating diatoms, and I'll show you some images of that, or eating other dinoflagellates and also feeding on other microplankton. Let's take a brief look at the food web, and on the left-hand side here we see the primary producers comprised of all these different types of phytoplankton. Of course, out in the ocean they'd be all mixed together, but I've separated them here by size and also abundance. So way more abundant are the smallest ones, cyanobacteria, pico-eukaryotes, then the flagellates and the coccolithophores, diatoms and dinoflagellates on top. The biggest ones can be eaten directly by animals pretty efficiently, things like salps and copepods, all the way up to fish and bigger things eating smaller things. But for these smaller ones, the primary predators on things like these small flagellates and the cyanobacteria and the pico-eukaryotes are other protists. So the animal grazing food chain and the things that are in it and some descriptions about them and the diversity there is coming in Module 7. Our focus is on the microscopic foundation here. So we've got most of the predators out there being protists, and that includes some of the phytoplankton themselves, which are also predators because they're mixotrophs trying to get nutrients by eating other cells. So we made these classifications before. The primary producers, here we've got the consumers, and then we've got the mixotrophs. Certain organisms within the primary producers are also predators. So the most abundant predators in the ocean are single-celled eukaryotes, protists, that are either heterotrophic or mixotrophic. They do most of the consumption of the primary production. So let's take a look at these. We'll just walk our way through some of these groups. Flagellates, it's not an evolutionarily coherent group. It covers a wide diversity of different types of eukaryotes, and there's a huge amount of diversity there. I'm lumping them together just because they have flagella and they swim around, but it does cover a wide range of sizes and diversity. And then we have the ciliates, which are a more coherent evolutionary group. Then we have the radiolarians and the foraminifera, also all related to each other within each of those groups. So we've got single-cell predators. Oh, how do they eat? They don't have claws, they don't have fangs, there's no mouth. So how do these cells eat? Well, many will swim. That's what these the flagella are for. They'll use a flagellum and beat that to move around, or the cilia will beat and move them, propel them through the water. But others, like the radiolarians and foraminifera, mostly drifting and waiting for things to bump into them. Without a mouth, what do? Well, they just wrap their cell membrane around the particle they're trying to eat, the prey they're trying to eat, and internalize it and do digestion inside the cell. So once they wrap it around and bring it in, they have a food vacuole. Other vacuoles in the cell have digestive enzymes that will fuse together and do the digestion inside of a vacuole that then fuses with the cell membrane again and releases any undigested material. What are they eating? Bacteria, phytoplankton. Some of these bigger protists are actually feeding on small animals, zooplankton. So things with flagella swimming around. Here are a couple of examples, some drawings of two different types of flagellates. You'll see the one on the left is a choanoflagellate. It's got a single flagellum and it's smooth. That kind of flagellum will propel the organism forward through the water. In this case, it's pointing down. It would propel itself downward in this image. The other one's got two flagella, and one of them's got these hairy projections on it called mastigonemes. You don't need to remember that. But it changes the hydrodynamics and how these things move, and these things actually sort of pull themselves through the water. So they're pulling water towards them, which means they're moving in the opposite direction. And here, these things are propelling this way, which means water's coming up. And they catch their prey. In the case of the one on the left, the choanoflagellate, they catch prey using that structure around the flagellum. So this is just a simple animation showing the process of phagocytosis, the means by which protists engulf their prey. And then once contacting a prey, it becomes ingested by wrapping it around and bringing it inside. So it's now encased in a vacuole inside the cell. Now we see digestive vacuoles with enzymes that merge with it to begin the digestive process in the vacuole. Digestion occurs, and then this thing can fuse back with the cell membrane and dump any remaining contents back out into the water. This is like just a little, we call it egesta. It's Protistan poop. So here's some examples of flagellates, just some light micrographs and electron micrographs giving you a sense of what they look like. And it's kind of a mixed bag. There's a whole bunch of these crazy little critters swimming around out there eating cells. They have some fun names. You don't have to remember any of these names, but things like Picophagus flagellatus, one of my favorites, Massisteria marina, Bodo sultans. There's another good one, Cafeteria ronbergensis. How's that for something that likes to eat? Cafeteria. And we introduced this guy before, the closest single-cell organism that's the closest relative to all animals, including us. This is where we came from, a cell similar to this one, choanoflagellates. And you see the resemblance there in the collar cells that you find inside of a sponge. Some examples of mixotrophic flagellates. So as we said, it's not just heterotrophic protists swimming around eating things. Some of the phytoplankton are in on the action as well, swimming around eating bacteria and cyanobacteria. And these are just some examples, and you can see they're kind of greenish, and that's because these things are phytoplankton. They just also happen to be predators as well. So dinoflagellates. So these are on the bigger side, as we said, really mixed bag. About 40 to 60% of the species have no pigment, so they're not photosynthetic. The others do, and some of those that do, sometimes they have it, sometimes they don't. And a number of them are bioluminescent. They also have some interesting feeding modes. They got these cellulose thecal plates. Makes it hard in some cases to actually engulf and bring prey inside. These have devised a couple of interesting feeding modes that allows them to eat things bigger than themselves. We already saw one of these modes shown on the left there, this peduncle feeding, where they're actually using it, in this case, to steal chloroplasts from their prey. Another one that's really cool, it's this pallium or veil. They secrete this pallium out and it wraps around prey. In this case, that's a chain-forming diatom, much bigger than the dinoflagellate itself, but it extrudes this pallium out, wraps around it, does its digestion there. Once it's digested, pulls that pallium back in. So lots of interesting ways to feed out there. So a little bit of video so you can see these things live. So here's a heterotrophic flagellate. Not really doing too much here, but you can see the flagellum a little bit, that whip-like tail beating around, and it would appear that they've eaten a diatom, that rod-shaped thing inside of them. Everything that eats has to. Everything poops, as we heard. Even single-celled organism. This is supplementary material from this paper by Okamoto et al. And here you can see it, folks. Protistan pooping. That's fabulous. Looks very satisfying. So those are flagellates, like some examples of ciliates. Cilia and flagella are functionally the same type of structure. The difference is things that have flagella just have one or two or a few. Cilia are just the same types of structure, but these organisms have a whole bunch of them that work in concert to create feeding currents and propel themselves. And they can be only at one end, sort of apical cilia like we see here on the left-hand examples. On the far right, you see one that's kind of hairy with cilia all over. Let's just take a look at some of those. So here's one with just polar cilia at one end, the apical projection. And you see these, it's looping now, and you'll see the feeding current as this little particle drifts in and then gets swept in, but then it gets cast aside. Either it wasn't something good to eat and it could chemically sense that and rejected it, or it just failed to handle it. This is how they move and also encounter their prey. I love this one. What we're seeing here is this rod-shaped structure. There is a diatom that's dead. It's just the silica frustule that's left. And as it's dying and decaying, the decomposers that we'll talk about next get in there and start growing, and the cilia is in there cleaning house. Then we have the radiolarians, also have hard parts, some with silica, some use strontium sulfate, but these are amoeboid types, and these aren't rapidly swimming around. They're just sort of drifting with their spines out and waiting for things to bump into them. So those are radiolaria. Kind of similar looking in their spininess are the foraminifera, and showing you an example here of a mixotrophic type because this one uses kleptoplasty, and you see those chloroplasts that it's stolen on those spines. These things also have a hard shell or a test. In this case, it's made out of calcium carbonate, and mostly they're microscopic marine plankton, but they can get quite large, benthic ones. They live in the plankton. They also are found in the benthos, and some of the benthic ones can get really big. And this is what the hard part looks like, the calcium carbonate test. And here's an example of one from these folks at UC Davis. And this is an example of a single-celled organism, that's this fuzzy thing, eating a brine shrimp, which it's captured there. Oh man, this is a single-celled organism eating an animal. Freaky. All right, so some key points from this video that I'd like you to take away from it is that most primary production in the ocean is done by microscopic cells, and so most of the consumption is as well. There are many diverse heterotrophic and mixotrophic protists out there eating phytoplankton and bacteria, and these include the flagellates, ciliates, radiolaria, and foraminifera. Some of these groups have hard parts, as we saw, calcium carbonate for the foraminifera, silica for the radiolaria, and cellulose for the dinoflagellates, and they're eating stuff by phagotrophy, this process of engulfment. We also saw in the dinoflagellates, what a weird, wonderful group, these other modes of eating that allow them to consume things much bigger than themselves.